I am very happy to be here. I was recently, I'm a member of the steering committee also of CML Advocates, and so I was recently in Serbia for the CML Horizons meeting, and I always felt that we have very good medical sessions with really great doctors that come and talk to us, and when I hear them speaking, I'm thinking to myself, does everybody know what they're talking about? And I know that unless you've had the background, it's very difficult to know exactly and follow the medical sessions. So I asked that I wanted to give this, what I call CML 101. It is not a medical session. I am not a doctor, and I will try not to pretend to be a doctor. But it's a session that hopefully we will go through some of the most important concepts that the doctors will be talking about later. And together we will try to make sure we understand what this language is all about and what these concepts are all about. So this is what I will try to do this morning. I want to say, because this is not the real Rising Sun yet, please feel free to grab your chair and bring it closer here if you need to. You don't need to stay in your assigned location. If you feel that you cannot see or you cannot hear well, please feel free to come and sit. This should be a dialogue. This is your opportunity, given that this is not the conference, to ask whatever question you want to ask to the doctors in advance to the lectures that we'll hear in the next couple of days. So this is what I call CML 101. And you will see that my slides they are the same slides I presented at CML Horizons. So you'll see that it shows the logo of them. And I wanted to make sure everybody knows who I am. I am the head of the Max Foundation. I am also the representative of Latin America. I was born in Argentina. This is where my accent is from. So I represent Latin America in the steering committee of the CML Advocates Network. I have a nursing degree, and I have a teaching degree. And I'm a member of the advisory board of the International CML Foundation. And of course, I am a caregiver in CML. I never thought in my life I would even hear what CML was, like all of us here. But I come to CML from the point of view of a caregiver of my stepson, Max, who was diagnosed with CML in 1988. This is before all these TKIs. And at the time, we looked for a transplant for him. We looked for a donor for 22 months. We never found a donor. So Max passed away in 1991, and he was 17. So it is my pride and my honor to be here with all of you. And hopefully we can help not only ourselves, but all the members of your groups back home. So before I go into this so-called not medical session, I wanted to make sure that everybody has thought about why you are here for this conference. What is it that you want to accomplish while you are here, okay? So this is my first question to you. Do you have any goals for this conference that you are about to attend? Has anybody thought of what are your goals while you're attending Rising Sun? Anybody that is brave? Some brave girl over there? Tell your name and your goals.
My name is Ka. I'm from Cambodia. I have set up the goal. I came here because I want to learn more from the other group around Asia, and I want to learn about the disease. I am the team of supporting group in Cambodia, so I can provide support to my group member, and also if any other track update, I can tell them, you know. And I wish to learn more so I can work on my group and make it more successfully and like the other group around the other country.
Great, thank you, Ka. Anybody else that has really thought about your goals? more or less the same as Ka, right? We all come here to learn. But if we want to learn, we have to be very specific about setting goals for ourselves. So this is my suggestion. My suggestion is that you really set up goals. So one goal, to learn about advances in treatment of CML, and you can achieve that goal through the medical sessions. Learn about others' experiences. So pay attention to the posters. If you are interested in something that somebody shows in a poster, make sure you find that person, ask questions, and take advantage of what others have done so that you can really apply that in your country. And you do that through the networking. So this is what we do when we have lunch and when we have coffee. Learn about advocacy. So the steering committee has prepared some really good advocacy sessions. And that's a very important part of your job as a leader of a CML organization. One important thing is to meet our speakers and industry representatives. So you have the opportunity, you have 2 experts, actually I think we have more than 2 experts through the Rising Sun Conference on CML. So make sure that you take advantage. They are here. We can sit with them at coffee, you can sit with them with lunch, you can ask them all the questions that you want. There's also, I think everybody knows Lucy, I think is coming from Novartis. There's another person from Novartis coming, Alice. This is very important. Take the advantage of making sure they know who you are, making sure you talk about your group and your members, making sure that you ask people for their business cards so you have their contact. This is very important. And then I believe when you go to a conference representing your group, you must produce a report for your group. Whatever you learn here cannot stay with you. So it's very important to take notes, to pay attention to what you're learning so that you can produce that report to your group. This is my advice. So now on we go with some of the medical terminology that you will hear. So this is the first thing that I think everybody should know. What is this thing that you hear so much about that's called BCR-ABL? And we want to make sure everybody knows what that is. So in CML, there is, everybody I think knows, there is this thing called the Philadelphia chromosome, right? And in the Philadelphia chromosome, what happens is this chromosome 9 and the chromosome 22, they swap, right? I think we all have heard that. So it turns out this break in the chromosome 9 is called ABL. The break in the chromosome 22 is called BCR. So people living with CML carry this gene that's called the BCR-ABL. So far, I'm going okay? Okay. So this is very important. So we talk so much in the medical sessions about BCR-ABL. This is what they're talking about, these cells that have this translocation of the chromosome. And this BCR-ABL gene is a protein that's called a tyrosine kinase. So that's the other thing that you will hear so much about, the tyrosine kinase, the TKI. And so this is what they're talking about, this, what really makes CML CML. Yes? So far I'm doing great. Okay, I just wanted to put this slide because I know that in the countries and through your members, this is a very important question that everybody always is asking, the causes of CML. And as far as I'm concerned, I'll ask the doctors, but the cause is still unknown. But here's what we know for sure, it's not hereditary. So people, if you have CML and you have a child, your child does not have CML, right? It is not contagious. So you cannot pass it along to a person just because you are close to them or because you are next to them. And this is the other thing that we know is that in Western countries, the average age of diagnosis is around 60 years old. But of course, we know this is not the reality in this region, that the average age is a lot younger, in fact, 36 or 38 years old. And it occurs more in men than women. I think we all know that. Anything you want to add? Good. I'm doing great. Okay, so we said that the BCR-ABL gene is this protein that's called tyrosine kinase, which I cannot exactly pronounce. So this TKI is a tyrosine kinase inhibitor, right? So it's a drug that prevents, inhibit means to prevent, to stop. It prevents the cells that have the tyrosine kinase, that have this protein. So that's what it's called, the TKI. And it's a very new type of medicine. So before imatinib was the first drug like that, very different from chemotherapy, right? Because chemotherapy kills the good cells and the bad cells, and that's why people get so sick. But these TKIs are targeted. They target the cells only that have this BCR-ABL. So this is what we call TKIs. Please say something. Explain with your words, since I'm getting nervous.
I think you're doing great.
Yes? Okay? Okay. And for you, maybe it's so normal, TKI, we talk about this all the time. It is a very, very new type of drug. So we're very lucky that the first type of TKI happened to be developed for CML. So there are 5 TKIs right now approved in some countries, not in all the countries, right? So imatinib, so then there's the names, and I will make sure that I ask the secretariat, make sure that everybody will get copies of this presentation, because these things are very difficult to remember. But we have to know the drugs and the names. You have to start trying to remember these drugs and the generic name and the brand name of the drugs. So imatinib is the same drug as Glivec. So Glivec is the brand name that Novartis gives, but the drug itself is called imatinib, and it's made by Novartis, right? Then there is dasatinib, and so you'll see all these drugs end with this same way, right? The names, dasatinib, imatinib, nilotinib. So dasatinib is Sprycel. So you'll hear Sprycel and dasatinib is the same drug, and it's made by Bristol Myers Squibb. Then there is nilotinib, that is also, it's called Tasigna. The brand name of nilotinib is Tasigna, and is made by Novartis. Bosutinib, the brand name is called Bosuliv. This is made by Pfizer. All these drugs are approved for CML in one country or another. So, and then there is ponatinib, is the newest one. It's called Iclusig in the market. The market name is Iclusig, and it's made by this company called Ariad. So this is one of the main things. As a leader of your group, you have to be very familiar with these names, and you have to make sure that you know all of these names, okay? And for some drugs, there are some other manufacturers making the same drug and calling it in different names. So imatinib has other manufacturers that are not Novartis that also have made imatinib, and it's called different names. Especially India has many sort of different versions of imatinib. So there are many types of imatinib out there. Okay. Okay, so this is also something that I want to make sure everybody understands, because I don't know that it's so clear. You will hear the doctors talk about first generation TKI, second generation TKI. In fact, some people, they say second gen. You'll hear them talking and say, and this is a second gen, and you don't know what second gen is. So they're talking about second generation drugs. So because imatinib was the first TKI, it's called first generation TKI, imatinib. So when you hear somebody talking about first generation TKI, they're usually talking about imatinib or Glivec. But then later on, the discovered, they made two different drugs, the nilotinib and dasatinib, which are Tasigna and Sprycel. And so people many times refer to these drugs as second generation TKIs. So I think it's very important to understand what that means when somebody is talking about a second generation TKI. Anything we should say about that? Okay, anybody has a question? I would like to hear lots of questions in this session because this is a session that is not official. So if you have any question about anything, this is your time to ask the questions, okay? And sometimes, oh, no, he doesn't. He's just waving and saying, hello, I'm here, Pramod. And I think sometimes people refer to ponatinib as third generation TKI. So it's because it is made later in the time. And these drugs are a little different from each other, right? They're not all the same. So I just want you to know when people say second generation TKI, it doesn't mean the same as second line treatment. We will talk in a minute about what is second line treatment, but this is just what means second generation TKI.
What exactly do you mean by generation? I mean, is second generation more advanced from the first generation or what? What is the difference between different generations?
Really only the time when they were invented. So for instance, nilotinib is chemically related to imatinib. It was invented by the same company, so they took imatinib and they changed the structure slightly to make it more potent. So they're actually related drugs.
So the generation is basically more potent.
No,
like
no,
third
it's
generation is more potent.
No, well, they happen to be, but the generation simply refers to the fact that it's newer. It's really about time. So dasatinib was made by a new company, and it's completely unrelated to imatinib. So they're not. When we talk about generations in people, we mean that they're the children of somebody, but in this case, that's not. It's just that they're newer.
I have a question about the definition of the inhibitors. Yeah, because the inhibitor for me is someone to live inside, but in here should be something different.
When you say inhibitor, because intrinsic to the activity of CML is the production of the BCR-ABL protein because of this translocation that happens, and this protein has got this tyrosine kinase activity. So when we give a drug, we are blocking the activity of this protein. So that is why it is called an inhibitor. The inhibitor of the activity of the BCR-ABL protein.
So to inhibit means to prevent, to stop.
I have a question for timing. As I understand, for the first generation is 1999, something like this, right? And the second generation is something 2005. Look like it's supposed to 5 years to have new generation. And the third generation is around 2010 or 11, something like this, right? Look like every 5 years we will have the next generation, right?
We can hope so. But it's interesting because, of course, the companies have to spend a lot of money to develop a new drug, so they have to decide, is there a need, is there some new advantage that we can get from new drugs? But there are new drugs coming that are just starting to be heard about, so maybe there will be a fourth generation, but you have to wait and see.
That is will be our hope, right? Hope is mean we will have the fourth generation drug soon.
Definitely there are many very interesting and important drugs in development for CML. So I think that you're right.
In some countries, they use a second-gen TKI as a gold standard. So do we see a better outcome or advantage versus imatinib as a gold standard? Thank you.
So the talk that I'm giving later today is on that subject, so maybe I'll leave it until then. But I think the answer, as always, is not simply yes or no. but wait and see.
So I also wanted to make sure that people understand what is an approved label. Sometimes we talk about the label indication and an approved label. So drugs are developed and tested, and then they get approved by the health authorities of each country. Typically, many times, the health authority in the US, which is the FDA, is the first health authority to approve a drug. So depending on how good the drug results are, the companies have to do these clinical trials and then present their data to the health authorities, to the FDA, and then the FDA approves them for use in the US. So the FDA only approves to use in the US, and every country has health authorities that have to look at the data that the company brings and decide if they're going to approve that drug or not. So in general, these approvals are very specific, and they're very specific to the results of the clinical trial. So a drug is not just approved for CML, and a drug is not just approved in all the countries. Every country goes through that process, and not always, the same drug is not always approved exactly for the same thing in all the countries. So if it's a new drug, the health authority might say that they will approve it only for those patients that are failing the treatment that is already in the market. So that is called second-line treatment. In general, when a drug is approved in a country, first it's approved as second-line treatment. That means that only those patients who have already had the treatment that is already been there for a long time, and that treatment fails the patient, because the patient doesn't fail the treatment fails the patient, right? So those that fail then can take this new drug. So this is what I was saying, second-line treatment is different from second-generation drug, okay? And many times in CML, especially when drugs get originally approved, they get approved for adults because CML is thought to be a disease of adults, and the clinical trials are typically done on adults. So this is very important because we know there's a lot of children with CML, and these drugs sometimes are not approved for children in our countries, right? So this is important to keep in mind because it's very important that you all know in your countries which drugs are approved for CML. And these approvals, if any of you have a box of one of these TKIs, and you look at this big paper that has all these little small letters that are very complicated, typically in that paper, this is where the label indication is there. And it will say this drug is approved for adult patients with chronic phase or accelerated phase. Some drugs are not approved for blast crisis patients. Some drugs are only approved for chronic phase or accelerated phase. Some drugs are approved for children, some drugs are not. And sometimes even the FDA and the European Health Authority, they approve a drug for two different things. So as advocates and leaders in our countries, this is the first thing to take note. I must go back to my country and I have to make sure that I know what drugs are approved for CML and what are they approved for. So only the drugs that are approved, the patients in our country can use. So this is very important. So when we talked about all these 5 TKIs, I don't know if there's any country here that has, in fact I know there's not any country in Asia Pacific that has these 5 drugs approved for use for CML patients. So this is a very important part for our advocacy. So this is what I was just saying. What if a drug is not approved in my country? And then we have a patient that we know, our friend, that needs some drug. And so we say we need access to this drug. Well, we can't. If the health authority has not approved, it is very difficult to get the drug in the country. So the health authority, in one way or another, has to approve this drug. And so in terms of access to these drugs, there are certain ways that are very specific and not very easy to successfully gain access. But obviously, a clinical trial provides some sort of access if a clinical trial is started in the country. Compassionate use is something that sometimes can be done by the companies, but you need the company to agree to provide compassionate use and the health authorities of your country to agree to accept that this drug is going to come in for this patient as compassionate use. And that is a complicated process. So this is something that we need to understand when we think that we need, and we know that we need all these drugs for our patients. But we need to make sure that somehow all these drugs get registered in our countries so that they're approved by the health authorities to use by the patients. And because it's very expensive to do clinical trials, not always clinical trials are done in our countries, and the compassionate use is very expensive, and the process of getting the approval for the compassionate use is very complicated. Sometimes it can take a lot of time just to get approval of one drug for one patient. So this is an area where we can make a big difference as leaders and as advocates. That's good? Okay. This is a slide I added. I didn't present about generics in CML Horizons, and then we had a session on generics, and all the people that were new to the conference came back and said, we didn't understand what is the big deal about generics. And so I thought that we need to really try to understand what is a generic and what is it that everybody now is talking about generics. So we said that after a drug is developed and tested, it gets approved, right? We said that. And so the country gives the manufacturer a patent. So the company spends a lot of money developing this drug. And this happens not only with drugs, with any product, right, that comes to the market. They get exclusive marketing rights for a certain period of time. Each country will say, we'll give you this patent for somewhere between 10 and 15 years. You are the only one that can make this drug and sell it in our country, right? So what happens is, because they're the only one, typically, and it costs a lot of money to develop, they're very expensive. And there's no competition. They're the only ones that can do it. But once that time expires, then other people can make the same drug. And usually the prices go down. This is sort of economics, supply and demand. If there's only one, there's no competition. If there's many, there's competition goes down. The price goes down. So what's happening now is, for the first time, the first TKI, which is Gleevec, imatinib, is starting to lose the patent in a lot of the countries. And so now it's legal for generic companies to make these generic versions of imatinib and bring it to the countries. We also have, and Dr. Hari can tell us more about it, but India is a country that allows company to make these generic versions of imatinib before the patent expired, for reasons that are very complicated to explain. So we know that there have been different versions of imatinib for a long time. They have come to some countries. And I think the big deal right now is that Gleevec is losing the patent in Europe. So now these drugs are coming into Europe, and the European advocates are very concerned because nobody exactly knows, is it going to be exactly the same? Is the effect going to be the same, right? And it's a little scary to know that somebody might switch my drug. And so there are some things about the generics and what's called the bioequivalence. Are they exactly the same? Is the active ingredient the same? And are the other substances that are in the pill the same? And are they going to react in the same way in people? So this is a very controversial subject right now. When we met a month ago in Serbia with the global CML advocates, there was a call from patients and patient leaders to all the stakeholders to put some guidelines that we would like to require as patients and as patient advocates, so that, first of all, that patients should not be switched for non-medical reasons, right? We don't want to be switched if it's not necessary. Patients don't want to go every month when they go to get their prescription, they get a different kind. So I think I asked Wei Meng to pass some sheets, some copies. There's a declaration that you can read of what the CML advocates are saying. That is the minimum requirements that patients want about these generic drugs. But most of obviously, the price coming down, I think we all agree is a good thing. We believe that there can be better access to these drugs, but we also want to make sure they're safe, these drugs. And I don't know if there's any comment that the doctors want to make about the generics.
With everything that you say, that, for instance, if someone switches to a new brand of imatinib and then their response doesn't seem to be quite as good, you'll be left worrying, was this going to happen anyway, and is this just a coincidence, or is this because the new tablet doesn't really work? Will there be new side effects because of the other things in it, or because it's not exactly identical? So it's going to make life a lot more complicated. So I think you have to have very strict controls.
Yes, I would agree with what David had to say, because we are worried about, especially in India, we have a huge number of generics that are currently available. I think, if I'm not mistaken, almost 14 different ones. So there is concern about it, and even as a physician, when I treat patients, I do use only certain specific generics for which I'm comfortable with. By default, we have had experience with those generics, not as part of a planned formal study, but it was efforts made by individual institutions like us to actually collate the data to see what the outcomes are for these patients. So we are always concerned about the new generics which have come in, but we are comfortable with using the older ones where we have had an experience with, and they have a little bit of a track record. But I agree that there has to be some sort of a formal studies, at least to have a certain credibility to its activity before its use.
Right, right. So physicians like Dr. Hari in India, they know the companies that they trust that make generics, but a generic comes to our country, and as advocates, the first thing that we need to know is what is the name, what company makes it, has it had tests of bioequivalence, so have there been any trials on this specific drug, and then we have to very carefully ask that our members and our patients are monitored closely to see what happens if these drugs are introduced in our country. So this is a very important topic at the moment. Yes?
I want to ask, when a health authority gives a license to a generic, for a generic, why don't they do the bioequivalence tests? Because if they do at that time, then that can be safe. Then why don't they conduct those tests?
So this is a good question for your health authorities of your country.
Okay, sure. I will ask, but I want to know why. I mean, you guys are there. You guys must have asked them, because you have experience. I don't have that.
This is a problem in many countries, that these tests are not done, and something comes, and this box sometimes it says approved by the FDA, which is not exactly true. What's approved by the FDA is imatinib, the imatinib from Novartis, but the health authorities sometimes don't ask for data. So Understanding the process of approval of drugs in our countries is a very important thing for our advocacy, and start pushing for bioequivalence tests and trials to make sure that, that the patients are safe. And so the CML community is really uniting right now. So, you know, a very important website, which is the CML Advocates Network website. There's a page on generics, and there's information that we are providing together about what generics are in each country and any safety concerns that we have. And so this is how, as a global community, we can work together on this issue. Yeah,
yeah, Pat, I'd like to follow up on that. The paper that Wei Meng gave you, it's a patient declaration on generics. Actually, the CML advocate sent all of us, or those that they know, email addresses of us, sent those for comments right after the Serbia event. And I don't know how many of us actually gave inputs, but judging from the way the Asia Pacific groups are engaging themselves in these kinds of surveys, on the third day of Rising Sun, on Saturday, I will be presenting the survey, and not many countries, Asia Pacific countries, get involved in these kinds of gathering of inputs. So, but they need our comments, the European groups and the American groups. So in case something like this happens again, please react to their emails. So just a reminder.
so it's interesting because it has become an important topic because the generics go to Europe, okay? So we, as advocates of the Asia Pacific region, need to make our voices louder so that our issues are important to the global community when they're happening, and not only when the problems get to Europe. But this is up to us to be part of the conversation and to be in touch with each other and speaking out about these issues. Dr. Razak.
Yeah, even if bioequivalent studies has been done on, for example, like one country doing a bioequivalent test, or even they did a clinical trials on generics for that particular population, does the company that produced this generic liable if, let's say, the drug is not safe or not efficient? Is there any global law that this company could be sort of like banned or made known to the rest of the world, that because if the information doesn't go to everywhere on the part of the world, people might continue buying these drugs. So who do we report to? That is the question. I don't know whether...
So there is, as far as I know, there is no global sort of entity that's looking at that. But this is why we as a global community are feeding information in the CML advocates so that we are writing about anything that is happening with these drugs at the moment. Yeah? Okay. Should we
Again, there is a library for generics at the cmladvocates.net website. Please visit it. They sent us emails again. Everything about generics is there.
And is there a session on generics later on? Yeah. So, all right. So we move on to the next subject. Okay. So treating CML in 2014. So there's a general consensus that when a patient is diagnosed with CML, they are put in one of these TKIs that is approved in that country for first-line treatment. So we already know what first-line treatment is. First-line treatment is a treatment that is approved in that country for people newly diagnosed. Because Gleevec has been around for some long time, in most of our countries, Gleevec is approved as first-line treatment, and most patients are started with CML on Gleevec, first-line treatment. In some countries, Tasigna is also approved for first-line treatment, and so some patients can be put on Tasigna or Sprycel, and so on and so forth. So we know that. And we said the labels vary, so in different countries, the treatment, the management of the disease is done in different ways. So, and we talked about this a little while ago, note to ourselves, I need to know in my country what drugs are approved, what are they approved for. If there's a drug that we think that our patients should have and we don't have, we need to go talk to the manufacturer of the drug. Are you planning to register your drug in my country? What is the problem? What is the process of registration of the drug? This is very important for our advocacy, so that our patients can have all the choices that are out there for treating the disease. So this is another thing that you will hear a lot about, drug resistance. And we need to make sure we know what drug resistance means. So sometimes the cells become resistant to the TKI. So the TKI is not able to inhibit anymore the BCR-ABL, right? And this happens sometimes because there's new mutations, right? So these new mutations don't allow the drug to fit exactly the way it was fitting before in the cell. So something we know, we hear a lot about, for instance, this is one mutation, the T315I mutation, because patients that have that mutation don't respond to any of the TKIs except for the newest one, that is the ponatinib. And so in the label, you will see that sometimes the second line label will say only for patients that are resistant or intolerant to the drug can take this drug. So this is what drug resistance is. And anything I say here, a lot of this is still unknown, but basically somehow some patients get resistant to one drug or the other, right? And because all the drugs are not the same, a patient can be resistant to one TKI but respond very well to another TKI. And this is why it's so important that our patients have access to all the TKIs that are in the market, right? Questions? No? Good. We know what drug resistance is. And this is drug intolerance. So when we talk about drug intolerance, we say that the patient cannot tolerate. It's causing very bad side effects. So some patients have more side effects than others. And so the way the treatment goes, you have to start with the first-line treatment, right? And then if you cannot tolerate this drug, then the health authorities will allow you to use the other drug that is approved for second-line treatment, right? So there are two reasons you can be allowed to use the second-line treatment. One is drug resistance, so not responding to the treatment, and the other one is drug intolerance, and that has to do with side effects. So it's very important that we make sure our members know that it's very important to tell their doctor about the side effects that they might be having. So sometimes this is a matter of the education that we have to do in our countries to our patients, to our members of our group, that when, if we're having a side effect, then instead of not taking the drug and not telling our doctor, it's very important to talk to the doctor and explain the side effects. And if the side effect is really bad and the patient cannot tolerate it, then it might be able to change to another drug. So you will hear people talking about drug intolerance, and this is what drug intolerance is. And I'm not sure if the doctors can tell us why some patients can tolerate and some patients cannot.
Every patient has got the unique way of metabolizing the drug and way of tolerating the drug. Sometimes there might be some deficiencies in some enzymatic process of activation or deactivation of the drug, which allows its concentration to be high or low. And there can be most of the time it's an idiosyncratic response. So one can never predict who is going to, but there are ways to do it, but many a time there is no way to say. So you just go by the clinical features, and if you cannot tolerate the drugs, you may re-challenge again, and if they cannot tolerate it. But if it's a life-threatening problem, you never re-challenge. but otherwise you reach out, and if it happens, then you'll have to withdraw the drug.
So when a drug is approved, this is what we are talking about, the approval of the drug. If the health authorities in your country said only people that are resistant or intolerant to imatinib can use this other drug, then this is where the intolerance is very important, because the only way to get access to that other drug is proving that the patient cannot tolerate the first drug, right? So this is why we talk a lot about intolerance and resistance. Okay, now we come to the most fun part of CML, which is the monitoring of the disease. And so I hope that everybody still has a little bit of energy, because so far it was easy. Now it gets slightly more complicated, but this is very important, the whole issue of monitoring of CML, and this is why we need to really try to understand it, right? So monitoring, the word monitoring means checking how the treatment is working or not. Huh? And so that means, is the TKI still inhibiting the BCR-ABL in the cells, right? Or has it become resistant? This is what we're trying to find out. Huh? And we need to check how deep in the cell is the response to the TKI. So this is why we are monitoring the treatment. And so I think all of you know more or less how we monitor CML. So CBC, or a blood count, monitors the hematological response to the treatment. So when you see, and you know probably when somebody is first diagnosed, the count, especially the white count, is very, very high. And you give a TKI, and immediately it's very common that the counts get normal. So that is the hematological response to the treatment. There is also the cytogenetic response and the molecular response, that is the PCR, right? And in this part, I will really make sure that the doctors are helping me, because this is quite complicated, I think. But let's see. Maybe Dr. Ross, you can tell us about this slide and a little bit describing the different ways to monitor CML. Do you mind?
So, I mean, hematological response, you've explained, the blood counts go back to normal, and if the spleen was enlarged, we can no longer feel it. So this is the way that CML was monitored 50 years ago, because that was everything that we had. The chromosome analysis, it's perhaps a little more complicated to explain, but all cells have 46 chromosomes, and when a cell divides, they spread apart. And so what we do is we take CML cells, usually from the bone marrow, we make them divide, and then we freeze them so that we can look at them and count them all up and see which one has the Philadelphia chromosome. That's a very complicated test which requires very highly trained scientists. So it's a little more sophisticated test. Typically you can only look at about 20 or 30 cells because it's very time consuming. And that will tell you whether or not the Philadelphia chromosome is present. And it will also tell you if there are other abnormalities outside the Philadelphia chromosome, which can sometimes be seen in people who've transformed to the accelerated phase or to blast crisis. So the chromosome test not only tells you is the leukemia still there, but it also tells you something about whether there have been changes in the leukemia, so that's sort of extra value. And then the molecular test, so Pat explained before that we make this BCR-ABL gene, so we can measure the number of copies of the BCR-ABL gene compared to the number of copies of a normal gene, usually ABL or BCR, and this enables us to get a percentage. But whereas with a chromosome test, we might look at only 20 or 30 cells, we might be able to look at 10,000 or 100,000 copies of a gene, so that we get a much more precise measurement and a much deeper measurement of the leukemia.
Okay? Let's try to see. So I think it's important, it's difficult to understand the science, but it's important to know that we must be able to monitor how this treatment is working in the patient, especially now that we have other drugs that can help the patient. So that the reason we're very interested in monitoring frequently how the patient is doing is to catch quickly if the patient starts becoming resistant to one drug, so we can do something about it. And so the most sophisticated and the one that measures the deepest response is this PCR. And this is why we hear so much about PCR monitoring. And this is, I think, the last very complicated concept to explain, the log reduction. So I will not even attempt. I will ask one of the doctors to explain to us this, what people talk about when they talk about log reduction. Mathematics, yes.
So I'll try to put it a little simply, because you have a huge volume of cells that are positive, or you have a huge volume of BCR-ABL protein. So when you calculate, when you give a drug, the volume, the amount of BCR-ABL transcripts, as we call it, they tend to come down. So it's difficult to actually go one-on-one and try to say that it is. So we try to say that in proportion to the previous volume, this is the amount of volume that has decreased from after starting therapy. So we calculate it in terms of log reduction. So when we say it is more easier for us to conceptualize when you say a log reduction, that so much proportion of the BCR-ABL has come down to such low proportion, so that we are able to give a value to it and also correlate it with certain other responses that we see, especially the cytogenetic response, as Dr. Ross just now told us. And we are able to correlate that if there is so much log reduction, then this is the amount of disease that is actually existing in the individual. And we desire a certain amount of decrease at certain time frames in order to predict, or even to say, okay, you are doing well and you are probably in some sort of a safe zone where you need to just continue your medication to sustain that response. Or let us put it another way, when you get into that safe zone, then your chances of progressing or your chances of having to move on to a second generation tyrosine kinase inhibitor are less. So these are just a way to actually quantify the decrease in the BCR-ABL transcripts over a time period.
Yes. So I think that the log reduction is now seen as a very important part of the management of CML, because, as the doctor is just saying, there's an expectation of what they have studied so much. What happens with a patient that has, I don't know, 3 log reduction after 6 months, or if they have 4.5 log reduction after 1 year, or something like that. The doctors have treated many patients, and they can tell which patients should continue on that drug and going to do well, or which patients should switch. So there's a lot of conversation about the log reduction and how fast this log reduction happens. And there are 2 set of guidelines for doctors that the doctors follow. One is the ELN guidelines, European Leukemia Net guidelines, about how to manage the disease depending on the response to treatment and the log reductions, and the NCCN guidelines, which is the US sort of version of the guidelines. And so I think it's important for patients to understand their log reduction, but I would like to ask the doctors, do you ever tell a patient, oh, you have 4.5 log reduction? Do you talk about log reductions with your patients?
Personally, I don't, because a lot of people don't have the mathematical knowledge, even in a country like Australia, where we have very good education, a lot of people don't understand the mathematics. So our test results come as a percentage, and I think people find that number easier to understand than a log reduction. So I think that I certainly tell my patients what their percentage value is, whether it's 0.1% or 0.01%, and most people have a better understanding of that value. So I think that most of my patients would know what percentage they have, and they would know whether it's stable or rising or falling. But I do tell my patients, you've now reached 0.1%, this puts you in a very good risk group, but I don't really talk specifically about the log reduction because a lot of people can't understand.
Dr. Haring?
Pretty much the same way. Yeah, it is very difficult for people to understand, and they get very confused and they get very concerned. It's got a lot of anxiety value, these values that I've shown. So I do discuss, as David said, the safe area where they are and what would have been desirable in terms of their responses and in terms of actual values. I rarely say that, I mean, I don't talk about log reductions at all because it is sometimes even difficult among the medical fraternity for them to understand what this log is. Right. So I would imagine that our patients cannot understand that. But if they do probe, yes, we do discuss that. Right. But otherwise, we talk in terms of the values, and in case there are any big fluctuation, then we voice our little bit of a concern as to, because we need to do more frequent testing at that point. So we need to justify why we need to do those frequent testing. So under those circumstances also, we discuss this value a little more in detail.
Right. Yes, Rod?
Maybe I can share a little. We cannot escape the term lag, and because I'm an engineer. For those of us which did not escape classes during our mathematics or geometry class, lag means exponentials of 10. That's why you consider it's 10, 100, 1,000, 10,000. The 3.2 lag is 32,000. But if it's lag 5, then it's 100,000. It's all in multiples or exponentials of 10. Because later on, I think in one of the slides of Pat, she will tell us that it's not the ultimate for our sickness. It's not actually lag 4.5. It has gone down to lag 15. So you could just imagine the sensitivity of that in terms of the exponentials of 10. So it's very hard, but it's a term we cannot go without. Lag means exponentials of 10.
You have a...
Well, my point of view is we can make it simple. Just counting the zero. For example, 2 log means 2 zeros after 1. 3 log means 3 zero after 1. So 100 times 2 log, 1,000 times 3 log. So just counting the zero. The decimal place is the same. If you have 1 zero, if you have 1 decimal place, this is -1. If you have, that means it's a 1 log reduction. If you have 2 decimal place, that means 1 zero after decimal place, and also 1 up, the 1 after the 0. That is 2 log reduction. So decimal place reduction.
Right. Yes, Dr. Razak.
What about undetectable? Because I've seen some reports says undetectable, but I suppose undetectable refers on the sensitivity of your machine. So is it a good practice to use the undetectable? Or because you might create excitement to the patient who are undetectable, but in the fact that it's your lab sensitivity?
This is a very, very important question and probably even more difficult for people to understand. But when someone gets the first test result which says 0, a lot of people think, well, now it's gone, it's finished. And that's really not the case. So we think that at diagnosis, when the leukemia is first discovered, there are about 10 million or 100 million leukemic cells. So if you have, say, a 5 log reduction, which would be the detection limit of even a very good laboratory, then that means that you probably still have, sorry, I've got the numbers wrong, so you probably start with many millions of cells. So at the limit of detection, you maybe have 500,000 to 1 million cells. So 0 actually means 1 million cells. Professor Goldman, who sadly died last year, gave a very good illustration of this. He told us the story about Nelson, the great British admiral, who was ordered to retreat in a naval battle, and he only had one eye. And he said, when his junior officer said to him, "Look over there, you should see the flag," he picked up the periscope, with a telescope, put it to his blind eye and said, "There's nothing there." So of course, if you look with a test which is not very good, you will see nothing. If you look with a test which is very, very good, then you will see the tiny, tiny little bit which is left. So when we say undetectable, it means nothing if you don't understand how good your test is. So everyone should understand that undetectable just means that this test on this day in this laboratory cannot see it.
I think this is such an important time right now, especially because we are trying to learn this, and this is very important for the lives of people living with CML. Yet you go to the doctor, and your doctor looks at your results and says, "You're fine," or says, "You're undetectable." So it is very hard for us to tell patients, first of all, the doctor says you're fine, you have to ask, first of all, what is your PCR? What is my PCR? And the doctor maybe reluctantly tells us the PCR number. Then we have to say, was the lab standardized? And what log reduction did I have? And so it is a very difficult moment, I think, where I think the medical community and the advocacy community need to come together to figure out what is the best balance between patients needing to understand what is happening with their disease and doctors trying to balance how much to tell patients.
I think that there's no general agreement on how to report these things. So for instance, even though our lab in Adelaide is one of the best in the world, when I get a result, it just says 0, and that's it. And it doesn't tell me any more detail about how good the test was on that day. So there has been no agreement in the scientific community as to how we should describe these results. So I think that people are thinking about that now, but no decision has been made.
Dr. Harry.
So the same issues, because in India we have people coming from various far and wide. So they come with lab reports from lab A on one occasion and on lab B from another occasion. It becomes extremely difficult for us to interpret that. So we, as a physician, I generally insist that you do it, if you're doing it in one lab, you better do it in the same lab. Don't come to me saying you've done it in another lab and then these are the values, because you cannot interpret that with conviction. So most of the time, so we need to identify certain specific labs, and the patient has to do it from that particular lab, because that lab itself becomes its own control, because to a large extent they would be doing the tests in a standardized way and the values that come would be comparable. Otherwise, you need to have the international scale for reporting the values. So again, when the value comes as 0, well, I do tell the patient, yes, it's nice to have this report, but the most important thing is please do not think about stopping the medication because I will like to see you down the line how this sustains, and then we could take a call subsequently. And then only after, I say about 2 years of having a sustained report, do I even talk about things such as, okay, there might be an option of thing, but we still have to look at it part in the framework of a trial or something like that. So we have to be very cautious about, because there is a sense of lethargy that sets in for the patient also as far as the treatment is concerned when they know that they are negative. It gives a false sense of confidence for the patient. It's important to have the confidence, but then you should not take it beyond.
Overly confident. Dr. Piti and then Dr. Rasai.
Okay, hi. I'm from engineering background with the test and measurements, right? So in the fields where I work is in metrology. So usually we have lab standards. We have committees sittings with scientific officers that decides that to measure some things, you have to have a lab standards, you have to have accreditations, you need to have a certification before you can say that, okay, you are certified, these results are good, before you can actually declare that results. So I would urge that for those who have access to the scientific committees or the scientific officers, that helps to set some standards. For example, like deciding, okay, in a lab with the IS standards, what are the criteria, how should it be done? Is there a list of IS lab in the world that you have these certifications? For example, like in the lines where I work, we have this ISO 17025 certification. So those lines with this certification, you are only allowed to supply such medical supplies or medical equipment to the industries. So for those labs with these standards, then only you are able or qualified to be able to treat CML effectively and such. So is that something that the world or the leading advocacy groups or even the medical society is driving towards too?
Those are very good questions. So I'm a pathologist. I'm employed by a pathology laboratory. And so there are metrological standards for a lot of the tests that we report, like the hemoglobin, for instance. There's a World Health Organization reference preparation of hemoglobin. So understanding this problem, the international community has developed a BCR-ABL international standard, which is recognized by the World Health Organization, but that was only made about 2 or 3 years ago. And I think it hasn't been used a lot yet. I think it will increasingly be used, but so far that was only invented relatively recently, and it hasn't been proven yet to improve the consistency of laboratory results. As far as the approval process, so Pat talked about the approval process for drugs. There's also an approval process for a laboratory test, and every country has different rules for what you're allowed to do. So for instance, in Australia, we're governed by NATA, for instance, the National Association of Testing Authorities, but NATA doesn't mandate that you must use a specific standard. So each country will have different approaches, and so far I think there is no approach, no country that insists that you must use the WHO standard, but maybe that will come in the future.
Thank you. So this is an area that is very new, that is really happening as we speak. It is an area that is very scary, I think, for the patient community, because the lives depend on understanding these tests and understanding the test results. And I think that we just need to have a little bit of an understanding that this is an issue that is really being settled as we speak. So I don't know that any of us have a very good answer to these questions, but we have to be aware that this is an issue that I think in 5 years, hopefully, we will be able to better understand. But there was an advocate when we were in Serbia who he showed the test result, and he goes to a private lab in his country, and the private lab writes the results, and this is a result on the IS. And he said, how do I know? I mean, how do I know? Is my lab just writing this? How do I know? There is no real, that we know, certification, some place we can go and find in a WHO this lab in a list that has been certified as being on the IS. So it's a very unknown thing for patients. And I think that this is a place where the scientific community and the patient advocacy community need to really have a good dialogue on this issue. No?
And if I could add to that, being on the IS doesn't necessarily mean that you have a good test.
Right.
Because the IS means that you have done some kind of process to show that your results are similar to other laboratories. But the other thing that matters is the accuracy. If I give you a result, today it says 1%. If you have another blood test taken tomorrow, will it still say 1%? Or will it say 5%? Or will it say 0.1%? So that is another question which is outside the IS question. It is how accurate, how much can I produce the same result for the same person every single day without it changing? And every laboratory has a range of results. So if the real number is 1, my laboratory will produce a value. So I know in my laboratory, if the real number is 1, the actual number will be anything from 0.5 to 2. In some labs, it would be 0.2 to 5. So my lab consistently produces values which are half to 2 times the actual value. In some labs, it's a 1/5 to 5 times. In some labs, it's a 1/10 to 10 times. And it's very important to understand that. And if as Harry said before, if you use different labs, then all of these different errors from different labs will be added together to make your test results almost impossible to understand. If you use the same lab all the time, then certainly the doctor who has many patients going to that lab will start to understand what the range is for this lab, because every lab will be different. So I think it's very important to find a lab that you trust and to stay with that lab as much as you can.
Thank you. And and we are trying to learn about this because we're going to be hearing about these things in the medical sessions, especially as we talk about the future of CML and the clinical trials that are being done now. And so I think that something to know is when somebody says MMR, I am in MMR, what does that mean, MMR? Major molecular response. So that is 3 log reduction, or 0.1. So that is a very good measure. And then they used to talk about CMR, which is complete molecular response, which now they call MR4 or MR 4.5. So this is the 4 log reduction or 4.5 log reduction. And this is the deepest that the tests of today can measure. So this is sort of the best response. And I have to say, I fixed this slide. So I have 0.0032 for 4.5, but in the slide before, I see I'm missing a 0 right here, 0.0032. So we got to fix that before anything else.
Just one short comment. I don't have the copy of the Boveno Declaration here, but as what happened during the CML Horizons in Serbia, the patient groups there, the paper of which is in your table, because of the comments about generics, they came up with a position. Now, it's still too early. We're just beginning the meeting. But what we can do, we will talk it out with the steering committee. If we can come out with a position on what PT said about a standards or a certification kind of stand, which we could, Rising Sun could give to an international body.
or at least things like we patients want to be tested always in the same lab. I mean, that's the minimum that the doctors are saying. So there are some things that I think as a community working with the advisors, like the doctors here, has come up with what are the minimum standards that we should be able to ask in this region for patients to be treated, that are treated with CML. I think that would be really great. Yes.
Okay, can I share my value? On the last two years, right? I still worry about my value of the PCR is around 2 point or 0.0 something. It look like it's bounce just only 1.5 years. And I just asked my doctor, why my PCR value not came down, right? And the first thing with I need the patient to keep, that is the PCR report for each time, because you need to compare what the PCR value with previous value, right? If the value is still the same, it will be good. But for some time when you test, it support bounce, right? If it's still bounce in the safe range, that is still good, right? But right now, for the last year, as I can remember, it still have just only log 4. But for this year, it's 4.5. For last year, I am sure my report just say just log 4, but for this year, just 4.5. That is, as I understand, right now, I standard have the deep, very deep, sensitive for checking, right? That is good for us. For me and for my member, I let them to keep the report of the value, because your doctor, it should be okay, you 0, but 0.0 something. But if it's still the same for the MMR log 3 for maybe 3 or 4 years, what that is mean? Just only log 3 for 3 to 5 years. Why it's not come to log 4? What that is mean? If I am your patient, right? It's supposed to, that value to go down. If your value still okay, log 3 for 3 years, and your doctor said that, okay, you are fine. But for me, not fine, right?
It's clear that some people have this pattern where every year the level goes lower and then eventually 0. But there are other people in whom the level reaches a certain point and then it just stays. So I have a number of patients who they just stay in a certain range all the time. We don't really know precisely why this happens. Obviously, it means that there must be leukemia cells which cannot be killed by that drug in that person. So obviously, if I had CML, I would want to be in the group that drops and drops and drops. But some people are and some people aren't, and there's no answer for it. I think that some leukemia must be easier to kill than others.
For last 2 years, right, I still on the log 3, just only maybe 1 year, and I asked my doctor, could I change my drug? Because I need to go down to log 4, right? Because I am not sure if I stay on the log 3 for maybe just only 10 years, is it safe for me or not, right? Then I think the doctor need to clear with the patient. Oh, okay, you are safe, you are okay with value, and how long you can stay safe, right? If you let your patient know, okay, now you just zero. What is the zero mean? 0.0.0 something, right? And how long I can stay with this zero value? For me, I need to compare back to the last three months or to the last 6 months. If the value still go down, that is safe for me.
So I think that if I can attempt to answer that, because I think there is no real good answer this moment for your question. But this is why the doctors, the key opinion leaders are coming with guidelines. They are actually investigating what happens in the long term with patients that the disease acts in a certain way and what happens in another. And when we look at the ELN guidelines, this is what the ELN guidelines try to do. And by the way, again, there is a patient-friendly version of ELN guidelines in the CML Advocates Network website that all of you should look at and all of you should be familiar with. So we know what the doctors are using as guidelines, and try to gently make sure our doctors know the guidelines, which is another problem, right? But it's interesting that we are talking about the log reduction and we're talking about MR 4.5 because the doctors will be talking about this. This is an important topic of discussion right now in the medical community. On the other side, we have our reality. This is one reality. They're talking about, is it 4.5 or 4 or 4 or 5 or what? Our reality. Most of our members are not monitored by PCR. Most of our countries don't have access to PCR. So we need to try to understand for ourselves why is it so important so that we can come up with actions to increase access to PCR treatment so that we can join and take advantage of the great research that is being done on CML. Because nowadays, the important thing is to have the tests available in our countries so that we can take advantage of these great drugs. And the members of our groups can live very, very long lives with CML. But so just to move forward a little bit, just want to talk about what is it that all the current clinical trials are doing. So I think the good news is, even though the results on CML are so good, doctors and researchers and drug companies have not given up on CML. Our goal is to eradicate CML. So a lot of the trials are done on how can we eradicate CML so that actually cure somebody. So that's a set of trials that are being done. The other set of trials is, can we, if we cannot cure the disease, can we achieve what is called treatment-free remission? So a few years ago, some brave patients that had been on TKI for many years said they were willing to stop and see what happened. So the doctors took the safest way, the safest patients, the patients they thought was safest, the ones that have had this very deep response, measured in very good labs for many years, and said, okay, stop and let's see what happens. And so they started measuring the PCR every month. And now it has been a few years, and I'm not sure that this is one of the discussions later on in the sort of the new trials that are being done. But within these trials, in a very controlled way, there are some patients who have stopped taking TKI and the disease has not come back. So a lot of different trials are being done in the world about this stopping treatment and is it safe and which patients should stop and which patients should not. So when we talk about, maybe the doctors will mention some of these trials, and so they have very specific names that are very complicated to understand. So I wanted to put here in the presentation some of these names so that when you come across some of these names of the most important trials on CML, you know what they are. So in this slide are the trials that were done when these new drugs were tested. So the first big trial was the IRIS trial, was the trial that Novartis did when they just came out with Gleevec. And they put two sets of patients, once on interferon, which was the drug that was used before, and some on this new drug, imatinib. And that was the IRIS trial. So many times you hear scientists talk about the IRIS trial, and now you know what that is. And then you have all these other names that are the trials that were used to test the new drugs. So this is very difficult to remember, but you'll have a copy of this, hopefully, and then you can always check back as a reference when you hear something like that. And so now we have these other trials, the ones that are measuring can patients stop treatment. And so one is called the TIGER, and some is called STIM, and STIM2. And I always get very confused, so I will ask the doctors when they speak later, if they name some trial, that they please explain to us exactly what that trial is trying to do. Because I hear people saying these names and I never know what they mean. The Euroski, and this is one of the biggest stopping trials in Europe right now. And Decline, I mean, they have very cute names, but they're very hard for us to follow what these names are. But the important thing is these are the areas where the scientists are doing a lot of research. And because most research is not done in our countries, we're a little stuck, right? Because we cannot take advantage of this research. So one thing that we struggle with in our region, in our community, is patients say, oh, somebody's stopping, so I also want to stop. And I think, it's probably true that in Asia, some patients would be able to stop and would be able to not relapse. But of course, because we don't have the trials, we can't do it yet. So as a community, this is another area where we have to work to see what is it that needs to be done to bring trials to our countries so that our patients can also take advantage of this new research. And so treatment-free remission is a new term that you will really hear. And so I wanted to give you some more resources to understand CML. These are all in the CML Advocates Network. So this is a website where we have a lot of information for patient advocates on CML. And you should be familiar with it. I think if your group is not registered, you should be registered in the CML Advocates Network and contribute to the discussions in the CML community. Okay? Any more questions? Yes, Donnie.
I'm asking in view of the patient. When we said the monitor, what is the object to be monitored? If for example, I know the CBC, the object of monitoring is the blood. So for patient to be abstract, the blood is okay. But if the monitor of the objections is the bone marrow, then the patient will be scared because to be objected, the bone marrow is very painful. Yeah.
So one of the things that we didn't say is that PCR can be done from peripheral blood.
Yeah.
So this is a very good thing about PCR.
That's what I mean.
Right.
Yeah, and we needed to let the patient know how easy or how difficult the monitor processing is. Yeah, if it's only by the monitor the blood, I think most of the patient is okay. but it's monitored by the bone marrow. Someone will say no.
Right.
Yeah.
So are you still doing bone marrow to your patients?
So I tell my patients when they're first diagnosed, in your entire life you should only have 2 bone marrow biopsies, one for the original diagnosis and one to prove that the bone marrow has returned completely to normal. And otherwise we would rely completely on the blood tests. Some people say you probably don't even need the second one, but personally I feel more comfortable having seen that the bone marrow is completely normal once.
Again, it depends upon where the patient is coming. If he were to come to an academic center, then we would be counseling the patient for at least another bone marrow because we are comfortable with that. We want to know what has happened to the cytogenetics and what is the cytogenetic remission, etc. So that is the only time we ask for a bone marrow. And maybe sometime when we ask for a bone marrow is when we are suspecting that the disease is not responding as well as we want to do, as well as we wanted to, or if we have a suspicion that we are going to discover some additional abnormalities which may predict how the patient is going to go 6 months or 1 year down the line. So these are all preemptive measures, but as David rightly said, we also do 2 marrows most of the time in our patients.
But I'd like to ask the doctors, with what Donnie said, yes, it's true. Some rural doctors, when you take into consideration the inconvenience for the very old patients and some of the young patients in terms of bone marrow, taking the sample from the bone itself, some rural doctors don't actually agree that we take just peripheral. So would it help, because I saw a slide showing, a PowerPoint slide showing that it's from a study that 95% when you take a peripheral against bone marrow, it's 95% accurate. So can we use that in our groups to tell the rural physicians, no, doctor, you could actually use peripheral because it's 95% as accurate as if you're taking it from the bone itself?
So a number of studies have compared the PCR test result and the bone marrow test result. So one study we did in Australia where we looked at about 400 bone marrow results and compared them with the PCR test, and they gave you the same information in something like 90% of the cases. So in the early stages when things are changing quickly, maybe they're not exactly the same, but later on, if you've been on treatment for a year and your level is low, the bone marrow tells you almost nothing on top of the PCR test. So we have very good evidence to prove that in certain situations the bone marrow will add no extra information.
Any more questions?
CML resistance to drug, it is a challenge. Is there any cause of resistance from which we can take precaution from it? Is there any cause of
You're
resistance?
asking what can cause resistance?
Yeah.
Fortunately, there is no real cause for developing resistance, but it is the intrinsic property of a malignant cells to evolve and overcome whatever treatment, and that is a dynamic process that is always happening, and the resistance you have is because of certain additional mutations or other mechanisms which will prevent the drug from acting as effectively as it should. So there is really nothing that you can do to prevent a mutation from developing or a resistance from developing. David?
I mean, the best way to prevent resistance is to take your tablets in the correct dose every day on time, because leukemia is just like the influenza virus or a bacterium. If you half treat it, then you leave the cells which are the strongest. So the best way to control an infection and the best way to control the leukemia is to take the best treatment consistently every day.
Thank you. So we're coming to the end of the session, and we all did very well, staying alert and trying to understand all this. I think I hope the great goal that we achieved this morning is to have this conversation among ourselves with our experts. So later on when they're speaking, they can say, remember when we talked about this morning, and remember when we said this other thing. And also we all already know each other, so they're not strangers. And when they say things we don't understand later on, we can raise our hands and ask questions because we already know the doctors who are going to have lunch with us, so we can feel free to come and have lunch with them and ask all the questions that you want to ask. So I really thank you for your attention, and I think that the boss over there has something to say. Thank you.